those of you who are just joining, we're happy to have this be quite interactive. So if you've got questions, feel free to ask questions as we go. I may or may not know the answer, but I've got the paper up next to me as well, and we can try and work it out if we don't know. This paper is good, but it's thankfully not too complicated. So hopefully we'll be able to get through the whole thing before we run out of time for the hour, and then there'll be some time for discussion at the end. How it sort of links back into memory decoding and the aspirational neuroscience prize and all those sorts of things. So yeah. Oh yeah, it's also going to be recorded. Thanks, Richard, for flagging that. And yeah, okay, I guess I'll take a sip of my tea and then we can get started. All right. Thanks for coming, everyone, to this this session of the Aspirational Neuroscience Journal Club, or the Memory Decoding Journal Club, or the Neuroscience of Learning and Memory Google Group Journal Club, whatever version of the club that you found the name for it. Today we're looking at this paper that's interesting. It looks at sort of engram formation in the motor cortex and in the dorsolateral striatum, how those two brain regions connect to each other, and what's going on down at a synaptic level during the learning of a particular motor task. So specifically this one, motor learning selectivity strengthens cortical and striatal synapses of motor engram neurons, by Fuu-Jiun Hwang as first author and Chun-Bi Ding as last author, some guys at Stanford. So let's get into it. All right, so yeah. We're going to be primarily spending our time in the primary motor cortex, which you can see there in this white little box, and its projections to the dorsolateral striatum, or DLS, and specifically the spiny neurons that's in that region. So why are we interested in this from a learning and memory perspective? Well, we know from previous work that the layer 5 pyramidal neurons, so the excitatory neurons in M1, are at least partially responsible for engrams underlying motor tasks, like running on the rotor rod or on the high beam, or any of the sort of behavioral neuroscience motor tasks we might get rodents to do. And we know this because, on the one hand, at a sort of correlative level, once you get animals to do these tasks, you see in the motor cortex things like enhanced dendritic spine formation, and delayed elimination of new spines in their motor cortex after these learning tasks, where the rate and stability of these things correlates with their performance on motor tasks. But there's also some cool previous work, for example, this one by Hayashi Tagagi in 2015, which showed that if you record which spines and which dendrites are changing, which structural changes occur during the learning of motor tasks, like running on the rotor rod, for example, during the training sessions of the mice, and then you use techniques to erase those new spines, things like optical erasure techniques using optogenetics, you can actually wipe out specific memories, specific procedural memories from rodents, at least to a certain degree with certain tasks. So really implying that something about these M1 spines, dendritic spines, that forms an engram that's critical for learning memories. So that's the M1 part of the region we're talking about. We're also going to look at the dorsolateral striatum, which is a major target of these M1 big excitatory pyramidal neurons, and which is also known to be involved in motor learning behavior. So DLS lesions or silencing these spiny projection neurons impairs motor behaviors. If you knock out NMDRs, so like glutamatergic signaling to a degree, you can prevent new skill learning. And the question is, like, what is actually going on with these M1 to DLS projections in forming these memories? So in this particular task, they decided to make use of this fairly hard behavioral task, which is a reaching task for a little like pellet, essentially. So it's like a little mouse is in the cage, gets a little slit, has to work out how to get its arm through and like reach out for this thing, which gives it a food reward. And as you can see here, it's pretty hard and it takes them a while to learn. So some mice, as shown in gray, never learn this particular behavioral task. So even after 8 sessions, they're still only performing at about a 10% rate. Some mice do learn. So they start off maybe at 20% or so, and they get up to 40, 50% sort of performance. And what's interesting about that group is once they learn, they do hold on to it to a certain degree. So when probed again after 8 training sessions at a week later, at the 15th session of the task, they're still performing well above the non-learner mice. So it's a quite hard procedural task that they use throughout this paper. And they use, in order to visualize what's going on in the M1 and the DLS throughout this paper, this particular kind of tagging technique for active neurons called targeted recombination in active populations, or TRAP. But what they have is a CFOS Kriya TD tomato mouse. But basically what happens is when that mouse is given an injection of this 4-OHT, so this chemical, immediately prior to any sort of behavioral task that it's doing, it induces permanent labeling of the cells that are active around the time of the injection with this TD tomato. So basically the recombination that happens is any of those cells that are active, they have like CFOS expression, and also it occurring at the time while they're getting this 4-OHT injection, will have a recombination event which leads to TD tomato expression. So those cells, for example, in its motor cortex, which are involved in learning the little reaching task, if they're active at the same time as their 4-OHT injection, then those cells will start to express TD tomato and turn red. And what's useful about that is that labeling will continue to be there even once CFOS expression stops, days later or something. So it's a sort of differential way of tagging cells that were active at some period of time around the injection of the 4-OHT. Okay, to get into a little bit more detail specifically of their training and injection procedures and labeling, they essentially had 3 different groups that they used. So they had control mice. So these were mice that were exposed to the training chamber, but weren't actually given any training on the task. So they're just there as controls, and they get their 4-OHT injections to label, cells that are active in the motor cortex on the first couple of days of training, and then they're perfused and stained towards the end. Then you have the trained mice that were given injections during the start of their training. So what this will do is it will label the cells that were active in the motor cortex during early training, when the mice is just starting to figure out how to do the task. And then they also get CFOS staining right at the end of the task. So what this means is with the early trap mice, you can distinguish between cells that are labeled with the TD tomato that were active during early training, and cells that had CFOS staining, which were active in the last sort of motor session just prior to the mice being killed and having their brains visualized. That's as opposed to the late trap mice, which have training, and then they have the trap performed on days 7 and 8 of their training. So once they're already quite good, hopefully, at the reaching task. And then they wait a week, and then on day 15, so a week later, after they've already been trained well, they have a final training session and CFOS labeling of those neurons that were active at the time. So basically the difference, early trap, you see with the TD tomato cells that were active when the animal's just learning to do the task. With late trap, you see the cells labeled that were being used after the animal was already particularly good at the task, hopefully. And with the CFOS labeling, as opposed to the TD tomato, you just see the neurons that were active in the reaching session immediately prior to the animals being sacrificed. That's sort of a little visualization of the difference. So you can see in pink the cells that are labeled by the trap TD tomato. In the bottom, you see the cells that are labeled in green immediately prior to the animals being sacrificed. And maybe some of those will overlap and maybe some of those won't. And that's sort of one of the key things they're looking at. Yeah, okay.
Hey, Ariel. When you say hopefully they would have learned the task, are you saying they weren't tested until they were sacrificed?
Not exactly. What I am saying, I'll skip ahead here, is that not all of the animals learnt the task. So if you look at the top right, you can see that in the gray line are the animals that were trained on the task, but never actually managed to like get pretty good performance at the task, even after 8 sessions of training. Whereas in the red line, you can see animals that actually learned to do the task properly and had sort of higher performance. So I'm saying hopefully just as like, you know, a behavioral neuroscientist who's gotten annoyed that their animals weren't actually performing at the task that you were trying to get them to do. For the purpose of the study, they're quite happy to just like contrast what happens in the learning group versus the non-learning group.
Okay.
But that's what I mean. Just that the like it reduces your numbers if not all your animals are capable of learning the task in question. Does that make sense? Does that answer your question? Yeah. Cool. Okay, so that's showing that. On the bottom, what you can see basically is the visualization of what they were specifically trying to do. So they were looking at motor cortex. I'll just finish this, Randall, and then you can go. And you can see in pink on the left, the neurons that are labeled with trap at any particular time when they did the labeling. In the middle, you can see the CFOS, which is the cells that were active and had these intrinsic CFOS expression immediately prior to the animals being sacrificed. And then you can see the merge of the two, where there's some overlap on the right. And I think it's the little green arrows in the bottom ones correspond to cells with just CFOS expression. The pink ones are just ones with trap expression. And the white arrows are those that have both CFOS and trap expression. And I point that out because they're going to use that labeling system a few times throughout the paper. Randall?
Yeah, so this is kind of an encore to what Oge was asking, what you then explained, because the kind of the follow-up question someone might have is, well, if so many of the mice couldn't learn the task, why are they using such a difficult task? And my assumption would be that's because we want to make sure this is a new thing they really have to learn, so they have to express a new motor learning. It's not one of their regular reaching behaviors or something like that, right?
I actually don't know, and that's a great question. Like, why use this as opposed to rotor rod or some beam task or something? I'm genuinely unsure. As far as I remember, they don't mention in the paper why they did that. I think that's a good suggestion that they wanted to make sure it was like genuinely a novel task. It could also be that they have other reasons why they're interested in this particular reaching task. It could be the lab's working on it. Could be it ties into other data they're trying to collect. I'm not sure. If anyone else has any idea why, that's a good question. All right. Also, actually, sorry. Another obvious thing is they do kind of want to look at what happens at like early phases of learning versus late phases of learning. And if you use a task that's too easy, then the animals can be trained up just during one session and they might get a lot better at it. So my memory, for example, with rotor rod is that animals get a lot better at the task quite quickly if it's more intuitive. Whereas if you've got something like this where like it's not intuitive and it takes them more training sessions, it gives you a better chance at looking at early performance versus late performance and what's happening. That's actually on reflection my best guess as to why. Yeah, but that's a good question. Okay, and this is just to flag the difference in cells that were labeled by trap and labeled by CFOS and what's happening over the course of the experiment. So basically what you're seeing here, double plus is the number of cells that were tagged with both CFOS and trap, and that's being divided by the number of cells that are tagged with trap. And basically what it's saying is, of the cells that were tagged with trap, how many of those also are expressing CFOS, which is another way of saying, when we look at the end of our little behavioral experiment at the number of cells that were active at the time, like during the motor task, just prior to the animals being killed, how many of those cells were also active at the earlier time points when we were recording which cells were active during the motor task learning. And what they find is that in the controls, obviously, it's only about 20%, which makes sense because there's no particular reason why the cells that were active in the chamber on day 8 will be the same cells that were active on day 2. Maybe some of those correspond to the motor things the animal uses to run around. And in the learner mice, what you see is a much higher proportion. So about 40% of those cells being active, which presumably means that the animals learnt the task well. They have this engram for how to do the reaching. And even though they learnt that on day 8, on day 15, they still use that same motor engram to sort of reach out and do the task. So those same cells are being reactivated. So you see that the cells labeled with CFOS largely overlap with the cells active with TRAP. And what's interesting is that you don't see that for early TRAP, which means that it doesn't look like the neurons that are used during the early stages of learning necessarily overlap to a large degree with the neurons that are active once the animals learn the task really well. So on day 8 of training. And you see the same thing for the non-learner rodents as well. Yes, Randall?
I think for that case of the early trap mice, it's also important to remember that, what was it? Which day? They received one more batch of late training, right? So in all of them, control, trained, and both those groups anyway. So if they didn't really learn the task in early training, how is it that they're doing that with other neurons? Well, they did get one more training session before that test, right?
Yeah, that's right. They do the motor task once more just before they immediately
some other
sacrifice
group
the
of
animals.
neurons was recruited for that newer learning at that point then. Yeah.
Yeah, exactly. All right. And yeah, I'll just show this and then Ken, you can go. This is just visualizing that in the learner animals, which have these success rates of above 35% or so, you see a distinct correlation between the number, the overlap in the CFOS cells and the trap cells, and the animal's performance. And this correlation doesn't exist for the non-learner animals or the animals that didn't learn the task. Ken?
So I had a quick question about the timing. Do you have any idea what the time window is for trap and the time window is for, I guess, CFOS, if they're going to sacrifice the animal and then do immunostaining? Is it like an hour or 2 hours or?
I honestly, I don't remember. I'll pull that up in the paper later. I don't know whether it's a day or an hour or 5 hours or something. That's a good question.
Okay. Well, while I'm at it, one more thing that's really striking to me, but maybe it shouldn't be struck by this, is that there is essentially no overlap between the behavioral performance of the learner and non-learner. It's like these are it's a bimodal complete distribution. It's like you're either never going to learn this or you're going to learn it pretty good. Am I reading that correct?
I suspect that's probably not correct, and that what they've done is they've really taken a sort of distribution and maybe split it into
so that's what I was thinking. But if you look at these error bars, the error bars do not overlap. So
yeah, I wonder. I mean, I'd like to see. I mean, maybe it's possible that it's bimodal, but I'm curious about seeing the individual traces of the individual animals and how their error goes and whether they've done exclusion. I mean, this is sort of just my bias from having run also hard behavioral experiments on animals where it's true that
you would expect that you would have to make a cutoff somewhere and say, okay, these are the non-learners and these are the learners. But okay.
No, you can. I mean, you can see to a degree. Sometimes there are tasks that really just some of them get and some of them don't. So it's not impossible. But also particularly with mice that are pretty dumb, their performance can be quite unstable and they can kind of get it and then forget, or they don't get it for 6 sessions and then finally they do get it or something. So I would also like to see the individual traces. I'd be surprised if it was really quite this clean, unless you're cutting it off, but maybe it is. Okay, so that's basically just the initial validation of their experimental setup to see that they can actually label neurons that are active at particular time points during learning the motor task. But then what they want to do is they want to go beyond just like entire pyramidal neurons down to the level of the individual dendrites and dendritic spines to see what's actually happening with synapses and connections. So what they do is, in this circumstance, they're interested in working out what's going on with these like late trap neurons. So animals that have learnt the task well, and that have progressively over time developed a stable ensemble of M1 neurons for performing the task. And basically what they're doing is they just want to do they're doing two-photon microscopy of those neurons like throughout the training period so that they can work out what's changing before and after the learning task. And they're doing that with two groups of animals. One is the trained mice, so mice that actually like learnt to do the task properly. And then their control group, which you can see on top, is where they're doing the same imaging every day for over a week of what's going on in their motor cortex, but they're just not training them on the task. And the way in which they do this specifically is they use their trap mouse population, but they cross it against a THY1 YFP mouse, which essentially sparsely expresses YFP in layer 5 neurons in the motor cortex, and I assume elsewhere as well. And what that is useful for is it means that now they can just generally visualize dendrites of neurons in the M1 in layer 5, even before they've done training or like trap or CFOS or any of those sorts of things. So it just means that like some of the dendrites in the layer 5 are going to be glowing yellow, and then they can contrast what they see in like prior to their behavioral training with what happens after the behavioral training by again looking at overlap between the yellow fluorescence and the red fluorescence from the trapped neurons. So to run you through what that looks like, you can see that before they're given the 4-OHT, so before they're trapped, on the top left, you can see that some of these layer 5 neurons are already expressing the THY1 YFP, so YFP. And in the top middle, you can see that there's a little bit of background expression of TD tomato, but mostly it's just pretty blank. So when you merge those two together, what you can see in the top right, which I've zoomed in on, is just like what some of the dendrites in the motor cortex look like. And you can see the like long strands of the dendrites, and then presumably the little spines as the little flecks you can see off the sides of those dendrites. And then what you can see on the bottom is what the same area looks like after training and after the trap procedure has been performed, where you can see now that robust TD tomato labeling. And if you merge them, which is in the bottom right, you can see the white, which is the overlap between dendrites that were labeled in green initially from before the training, and the those neurons that were active during training. And you can see they're flagged in white there to sort of visualize the overlap, which just is validating that they can do this labeling of the dendrites, and they can do the labeling of the spines that become active during the training sessions. And specifically what they want to point out is that over the course of training, so they're looking at, this is just looking in the brains of the animals that were trained and successfully learned the task. They're distinguishing between neurons that were trained and then trapped. Animals that were trained, neurons that were trapped. So neurons that they established were definitely part of the engram that served the animal's behavioral performance, and other neurons in M1 that weren't trapped, so that presumably weren't part of the engram for the animal's behavioral performance. And what you can see here is the spine density changes over the course of the training. We're taking the pre-training period as baseline. And what you see is this initial spike up in the density of these trapped neuron spines during early training that then comes down to back to about 100%, so back to the same level, whereas the non-trapped dendrites actually, spines actually reduce over that period of time. So it seems that like there's a reduction in the number of spines, an initial increase and then reduction in the number of spines, but the spines that are selectively spared are those that are involved in learning this motor task. And in another way of visualizing it, if they're looking specifically at like spines that are newly formed and then whether they survive over the course of the imaging window, you can see there's very much selective sparing of the new spines that are trapped and thus that have been involved in this procedural task. They show this in like a lot of different ways, but these are really the two key figures that make it the clearest. And they use that to say that our in vivo imaging data demonstrates that M1 spine plasticity during motor learning preferentially occurs on dendrites belonging to trapped engram neurons that are activated in well-trained mice during motor reaching tasks. And I think they demonstrate that pretty well. I'll just make sure everyone's still on the same page before I go on, because this is sort of key initial parts before we get to more of the structural stuff. All right, getting some thumbs ups. Okay, so that's cool. But that's really all just been M1 so far. And this paper is supposed to be about how does the M1 interact with the dorsolateral striatum. So we've got to actually get to the dorsolateral striatum. So basically, this is this exquisite diagram here. I say as someone who's also made some very bad diagrams over time, is showing the how the M1 neurons connect into the dorsolateral striatum. Just showing that they project down to the spiny projection neurons here. And what they do in this particular experiment is now, instead of just looking at how the M1 neurons are naturally active during the motor tasks, they also want to have a way of like artificially stimulating those M1 neurons that are either involved in the, that they're part of the engram for doing the reaching task, or they're just random M1 neurons or whatever. We'll get into the variations. And they do that by optogenetically inserting the by optogenetically inserting the channel rhodopsin 2 labeled with M-cherry. So now they're going to, basically, when they shine the light on them, they're going to become active and send signals to dorsolateral striatum. Well, I just realized, this is sort of an aside, channel rhodopsin 2 is sensitive to blue light, isn't it? Not red light. So I think my red laser there should actually be a blue laser. So apologies to those of you watching. M-cherry is tagged with red, but the channel rhodopsin 2 should actually be a blue light. So anyway, this just means that now they can selectively turn on this population of M1 neurons, and they can see what happens in the dorsolateral striatum when they turn these guys on. So again, they're going to be doing this in control mice and in the early trapped mice and in the late trapped mice. And they want to see what influence the M1 has on these dorsolateral striatum neurons. And what you can see in the bottom here is basically just how they're responding when the M1 is activated and the patch clamping is happening on some of the neurons in the dorsolateral striatum. This is done in ex vivo experiments. And what I find fascinating is like just how responsive these DLS neurons are to M1 stimulation. So even in the control mice, it seems like 60% plus of the neurons, you can see these postsynaptic currents in when you shine the light on the M1 neurons, going up to basically 100% of neurons in the late trap mice. So that's on the left. In the middle, you can sort of see the like strength of these current flows. And on the right, you can also see like another visualization of the strength of the current flows, the EPSCs evoked by M1 stimulation. And so the key thing they want to point out is that in the late trapped animals, you see even stronger EPSCs elicited than in the control and the early trapped mice. So when they've established this really well-defined engram for the motor reaching task, that induces this change in plasticity from M1 neurons onto DLS neurons, as recorded here, almost a doubling in the currents that they're able to observe. Ken.
So I want to make sure. So the number of cells in M1 that are being stimulated at a single time would probably be quite large.
Yes, that is true.
Okay, and so that would raise the statistics for at least one of those having a connection to a dorsal striatum cell, and so maybe that explains why the responsive neurons are so high.
Yeah, I mean, this might be totally normal for cells in the DLS. I'm certainly not an expert on the electrophysiological properties. I'm just used to like seeing hippocampal slice recordings or like recordings from elsewhere where you often get like very sparse connectivity and activity. And I just looked at this and I was like, wow, that's really like impressive in terms of.
I mean, it's just like for instance, the paper we did a couple weeks ago that was activating a single cell and listening to cells that might be connected, and you would get stuff like 2%.
Yeah.
Right. And so now if you were activating 100 cells, then you would actually expect almost 100% to respond.
Yeah, I guess maybe I shouldn't be so surprised. It's true. They're recording from an entire engram, which again is just like a small subset of the total M1 neuron population, but is still like a very large number of cells compared to doing those single cell stimulations. Okay, so they've established plasticity in the connection between M1 and DLS, but they want to look at that in more detail. And they want to actually look at the dynamics rather than just like the strength of the connections. So they also do some calcium fluorescence imaging. So what they do is they insert this calcium indicator into the dorsolateral striatum, so into these SPN neurons, so that when they do the optogenetic stimulation of the motor cortex, they can actually see which particular spines become active on those dorsolateral striatum neurons, on those SPN neurons. And again, yeah, it's 450 nanometer stimulation, so my bad from before in terms of showing should have been a blue laser. And what this is going to let them do is basically when they do their optogenetic stimulation of M1, they can track which spines are becoming active. So you can see on the top, there's a little trace where after each stimulation at 2 and 4 time seconds, they can see fluorescence. And in the bottom, you can see one that's not quite a stronger response, but still clearly above baseline in terms of fluorescence changes. So this is what they're doing for their window for all the spiny neurons they can see in the DLS. And so to point out what they find, if you look at H, so the graph in the middle, you can see that the number of spines that they're able to visualize in early trapped animals and in late trapped animals is roughly the same. But if you look over at G on the left, you can see that the number of active spines, so the number that are fluorescing during the stimulation, is much higher in the late trapped animals than in the early trapped animals. So it seems to be the case that animals that have formed this engram for doing this reaching task, if you look at the late trapped neurons, so if you look at the DLS neurons that are downstream of that engram, that you see much higher activity in those spines when you have this optogenetic stimulation. So there's like much greater functional connection between the M1 and the DLS as visualized by active spine densities. That's essentially all they're pointing out. It's just that you, again, get a visualization of this plasticity. So finally, that leads us into, oh, Ken.
Sorry.
No, it's good.
On G, comparing G to H, they're both in spines per micron in terms of density. So the late trap in G versus the late trap in H, if we compare those two, it's the active is about 20%. So this is saying that 20% of the spines were active spines.
I do think that that is what they are saying. They're saying that 20% of the active spines that they visualized in the DLS became active at some point during their visualization, as
Okay.
opposed to
That's a lot.
almost. Yeah, this is what I mean by like it's like a lot of activity, and the same with the connections previously. Yeah, again, like I don't know what to expect from the DLS, but I also thought that was quite a lot. So yeah, finally we are going to the, how this relates back to aspirational neuroscience and memory decoding, and like actually looking at stuff from structure, because the last figure in their paper, the key thing is focused on the spatial clustering of these spines. So not just like their plasticity, functional properties, but where are these actual synaptic spines from M1, these synaptic connections from M1 onto these DLS neurons. So basically, their previous experiments have established that there is this plasticity between the M1 and the DLS, and they're asking now, but what about structural connectivity? So what they're going to do is they're going to examine synapse density and specifically synapse clustering in these early trap animals. So again, those that were trained and injected early on during the training session. In late trap animals, so those injected on day 7 and 8 to label the neurons that were active towards the end of training. And they're also going to use this Thai-1 channel rhodopsin 2 animal as a control. So that's not the same as the Thai-1 YFP I mentioned before. Now, instead of just intrinsically labeling some population of random layer 5 neurons, they are also going to be able to optogenetically stimulate some random population of M1 neurons in these control mice to see what happens with synapses and synapse density and clustering if you're just like randomly picking a subset of neurons in M1 to stimulate and then influence the DLS. Yeah, that's just reiterating what they said there. So what they're going to do is they're going to repeat, as previously shown, optogenetic stimulation of tagged M1 neurons in these groups. So whether that's the early trap neurons, the late trap neurons, or a random population of neurons in M1, and they're going to observe the active spines and their locations. And just finally, to give you a sense of the activity levels, as sort of Ken was just talking about before, if you look at the active spine densities, you can see it's pretty high in those late trapped neurons. It's pretty low in the early trapped neurons. And for the Thai-1 channel rhodopsin 2, so just like the random labeling of some M1 neurons, you still get pretty robust response from the DLS neurons when you're just stimulating a random population of motor cortex neurons. So that's what's indicated there in green. While meanwhile, on the right, you can see the spine density remains pretty similar in the DLS for all of those different groups. Randall?
Yeah, and just a curiosity here. So can you explain why the Thai-1 channel rhodopsin 2 mice in E, why they seem to be have a somewhat higher percentage than the early trap? Is it that the early trap has to do worse for some reason? I'm not quite understanding the statistics here, why that's not similar to the Thai-1 channel rhodopsin 2.
I also am not entirely sure. I don't know whether it's because, for example, it could just be that the Thai-1 channel rhodopsin 2 is labeling a larger population than that which gets tagged by the early trap neurons.
Right. Yeah, that's probably it. Yeah.
Because it's not controlled in terms of like numbers of cells in M1.
No, that makes sense.
That would be my best guess, but I'm not entirely sure. All right, and here basically is the key thing that they show in terms of the clustering. So they've got a statistical representation, which I'll show in a second, but I do quite like these visualizations. So again, what they're showing in the different colors here is just the active spines that they could label, where it's those active connections between the M1 neurons and the neurons in the DLS. And what you can see on the top left, so the early trap, is just showing that there's only a few active spines that they can visualize in these DLS neurons. So there's not that much of an active functional connection between those populations. If you contrast that to the top right figure, so the late trap, you can see that there's a lot of active spines, as previously established, but also that the spatial distribution of those active spines is quite clustered. So you can see like most of those spines are located pretty close to each other, so within like 10, 20, 30 micrometers or so. And that is in contrast to the bottom one on the left, where you can see that there's a lot of active spines, so a lot of active connections between M1 and DLS, but there's a much more random distribution of those active spines. So they're not clustered up in the same way that those late trap spines are. Randall?
And I believe if I read the paper correctly, they said that they calculated the clustering for B and C, but they didn't calculate it for A because there simply weren't enough to do that.
Correct.
Is that right?
Yeah, they didn't even try for A. But they did calculate it, which I will show here, for example, where they do a nearest neighbor distance. So they're looking at taking the spines and seeing how close they are to other spines. And then they do their stats by like randomly shuffling around the spines and seeing the distance to get their 95% confidence intervals. And you can see specifically in the late trap animals that there's just this big spike in proximity of around, 5 or 7 micrometers or so. So most of the spines are like tightly clustered together, as opposed to in the thigh 1, channel rhodopsin 2, where they're just like broadly distributed throughout. And this is sort of like, as far as we're concerned, the key takeaway from this paper, that you're seeing this spatial clustering of these engram projections onto the DLS. And this is it. This is their final sort of like fun little visualization, just to show what they think is happening in these animals, where they think that, initially you've got these connections between the M1 and the DLS, where there's the projection axons, and there's a bit, but not very much in terms of active connectivity between the two. And then over the course of motor learning and training, you get the formation of more of these synaptic connections, and specifically that they do so in a clustered active sense in close spatial proximity. That's what they think that their set of experiments demonstrates. So to get into the discussion, before I open it up for just general chat, they, I think, show quite well that this M1 and DLS engram ensembles are useful for these well-trained procedural memories, and that these are like stable engram populations. So they show that those cells that are recorded on day 7 and 8 of training using the trap mechanism are the same cells basically that are active a week later when they visualize using CFOS. So there's some stability, at least for a week's time, in the motor cortex in terms of the cells that are underlying that representation. And they say that like maybe this is what is helping to serve stereotyped actions. So they talk, for example, about the fact that as the animals get better at the task, they seem to use much more like stereotyped hand movements to sort of perform the task, which I guess seems similar to like any sort of procedural tasks, where you see them become more and more stereotyped as humans become better trained at them. They note that it's quite interesting to see this spatial clustering of the M1 engram neurons onto the SPN neurons, which they suspect, I think quite reasonably, helps with sort of superlinear integration of outputs from the M1 onto the DLS neurons. So ensuring presumably action potentials in those SPN neurons. And then one thing I found very sneaky is that in the conclusion, they have this like key throwaway line, which I would have loved to have seen some data in the supplementary material on, which is they say that preliminary chemogenetic silencing of these M1 engrams did not significantly affect performance of trained mice. Now that's on the one hand, that's pretty weird. So they've got this whole experimental setup where they're like showing the development of these robust M1 engrams, and then they're saying, but actually they don't matter, because if we like chemogenetically silence them, the animals can still perform the task just fine, which is that's like pretty weird. And it also is interesting because you see some contrast between this particular chemogenetic silencing not doing anything and the like previous paper that I mentioned right at the start in the introduction, where they show that if you optically erase these sorts of N1 engram spines, that you can significantly reduce animals' performance at these procedural tasks. Now, I would just point out that like one of the key differences, though, between their experiment and this experiment is the erasure experiment happened in the rotor-rod task, where learning had happened only over 2 days and then was erased on day 3. So potentially, they did a better job of capturing all the spines that were involved in learning that task. Maybe the task hadn't been transferred to other cortical regions or brain regions as of yet, as opposed to in this task where the training happened much more extensively over 8 days. And then I don't know when they tried the chemogenetic silencing, but the task itself was still seeing robust performance at day 15. Randall?
Yeah, I mean, you kind of went into what I was going to say because, this totally jumped out at me as well when I got to the discussion section. But then they kind of did the sort of throwaway, well, it's probably been copied in various forms in other areas of the brain by then. And so, yeah, the time duration kind of covers that. Maybe. But again, they didn't really explain it. They just made a suggestion there.
Yeah. Yeah, I just thought that's certainly one of the most double takes I've ever had while reading a conclusion to a paper, where I was like, hold on, that's like, you did a whole set of additional interesting experiments and it's just like not mentioned, no supplementary data or anything. Ken?
Yeah, I was thinking that was interesting as well. And I'm wondering what is known about motor learning consolidation. I remember reading a bunch of things a long time ago that made me think that motor learning happened in the basal ganglia and the striatum primarily, and then it was transferred to cortex. This is very loose because obviously striatum is getting a whole bunch of input. And I think they said something earlier in this paper that if you wipe out the learning in striatum, that mice don't learn this task at all. Or
yeah, they say that if you do lesions of the DLS, or if you remove NMDARs, so you prevent like the NMDA activity required for plasticity, that you get impaired behavioral performance. I don't know whether it was specifically for this task or other tasks or motor tasks in general, or with what time conditions. I mean, this is the thing, right, where we're talking about like the time courses really matter, the particular procedural tasks matter, and I'm not on top of all those details, but it did make me curious.
So how much does this bit undermine the significance of the paper, if at all, do you think?
I don't think it undermines the significance of the paper. Well, I guess insofar as like, I think it's well established that you get engrams formed in multiple locations at the same time and that subserve the same memory. So you like think of all the like episodic memory, conditional fear conditioning stuff, where they show simultaneous formation in the hippocampus and in cortex, like anterior cingulate, and that like what happens over time is which becomes responsible for recall events when induced by natural cues switches from hippocampus to cortex over time. So it's not that I become suspicious that they actually haven't found the engram if silencing this engram means no behavioral expression. Yeah, I guess it doesn't undermine the paper. It's more just like fascinating that it makes me wonder like, well, hold on, like where is the engram or the set of engrams, or like what is the full like set of brain structures that underlie this like particular memory?
That's exactly what I was thinking as well. It made me wonder about the definition of an engram. When do you call something the memory engram of this, or how do you draw the boundary? How fuzzy is that boundary? Is it groups of things that make like clusters of engrams that belong to the same thing, or what?
I have a memory. I think there's a Tonegawa paper, like something from the MIT lab where they do sort of memory tasks. I might be getting this totally wrong, so apologies. I'll try and find it later. But it's something effective, like they do motor tasks, and then they try and do almost whole brain visualization of like structural changes after the task. I can see Ken nodding away, so he can jump in at any point. And what they basically find is like changes all over the place during a particular procedural task. During a particular memory task. So as a guess, that would be like you do something like conditioned fear conditioning, and you see changes in hippocampus, but also changes in cortex, and also changes in amygdala, and like changes elsewhere. And like maybe each of those is redundant, or each of those adds slightly different information to the memory, or I'm not sure. My guess is it's pretty broadly distributed and maybe partially redundant. Yeah, just Randall.
Yeah,
go for it.
Yeah,
We're pretty open here. I've got one more slide afterwards.
Okay.
But
This is a question to Ken, actually. Now, having seen this paper and what you can do and what you can recognize, just looking at the static snapshot and all that, can you imagine, Ken, a way of taking what this paper has done to something that would win the memory decoding prize?
Okay, well, this is a question that I wanted to ask Ariel, because before I would go there, I would have to kind of understand a little bit about what the representation is in M1 and in the striatum. Is there kind of a any type of an understanding of what that representation would be?
Again, I don't know the answer to what the representations are that people expect in the DLS. I actually learned a lot about DLS stuff while doing this paper. I have not previously looked a lot into striatal representation or activity underlying procedural memories. So I'm really still not sure, having read this. Maybe, but getting into that question though, Randall and Ken, let me just quickly run through the last slide and a half, which is sort of bears on that, and then we can just have open discussion. So in terms of other sort of like remaining questions I still had after this paper, there's a few things in a like technical procedural sense that I wanted to flag that kind of annoyed me because I couldn't find them easily. So one thing I was a bit confused by was what was the actual window between when they did the training in these animals and then they did their optogenetic stimulation in an ex vivo sense or an in vivo sense. The only thing I could find was a throwaway line somewhere about how they confirmed that they could see the channel rhodopsin 2 expression 8 weeks after they did the trapping, which is a long time later, but I couldn't find what the actual training window was, the window between those two was. A question I had, which is like, they mention that the synapse clusters they see primarily occurring at 50 to 70 micrometers away from the soma of these animals, of the SPN neurons. And I just wonder if there's a particular reason why it's like that location. Is it just like that's where most of the spines are? Or is it like that's the ideal location for doing...
Didn't they mention these were on apical dendrites?
I
recall that.
Yeah, I think so.
Yeah, so it's probably on the apical tuft at the end there.
But there was still a lot of dendrites. Like from memory, they show that like the tuft exceeds like is like reasonably long. You could conceive it to be elsewhere, I would have thought, on the tufts. But maybe that's just the I'm doing a bad job of phrasing my question. I'll come back to that later. It's not actually that important. I was just curious. And then there's, finally, and this gets back to what we were just discussing, the implications for memory decoding, where like my takeaway from like something that might be useful from this paper for considering memory decoding is just that these I don't know if this is the right term. I'm going to call it intra-engram in that it's, part of M1 and part of DLS, but it's the same procedural memory that there's an engram for. So these intra-engram synaptic connections between brain regions seem to be forming these distinct spatial clusters. And my question would be that if you train these animals up on like a set of different behavioral tasks, so like the reaching task, but maybe also the rotor rod, and maybe also high beam, and maybe also some weird running task or whatever, would you be able to identify distinct clusters of these synapses from M1 neurons onto DLS neurons. And if there are the formation of these distinct synapses, would it only be the case that you could identify them using active methods, like the fluorescence that we saw in this paper, while the animals are still biologically active? Or would it be possible to identify those clusters somehow using tracing studies after the fact? And that's it. That's all the material. So it's fully open now to anyone to chat.
I mean, I don't see why you wouldn't be able to see this from tracing data. If you knew what upstream neurons were part of the M1 engram, then you could look and see if their synapses are clustered. I'm kind of wondering, you know, I appreciate that clusters can do things, and so they shouldn't be ignored. You know, it's an amplification at the very least. But I'm wondering if clusters, like if they didn't find clusters, if there literally was no clusters, that wouldn't change my mind about this paper. They found that they had more synapses, and they found that they were stronger.
Yeah, I mean, it would.
Thing was just that's earlier on when you then switched to the clusters and said this is like the main thing that they found. I felt like, yeah, before when they were counting them, that was also just as valid in a sense. Yeah. Fair
enough. I guess I mean, I don't think they would agree necessarily. I think they'd agree with you too, that like this isn't necessarily the main thing. But I was just wondering, sorry?
Well, I mean, there's a lot of enthusiasm out there in the literature for clustering. It's like fundamental neuroscience. You know, does a neuron is it well characterized as a single node in the network or not? And so these papers really kind of push that out. But if you look at the mechanisms, at least in my mind, it's kind of if there is any kind of local calcium boost, I would expect that LTP would naturally form clusters. The other thing is that if I have an axon going by, it often makes two synapses because the dendrite can reach out twice to the same axon. And so I'm wondering if anybody has actually kind of looked in and said, you know, because a lot of times I read these and it's like, oh, it's actively clustering. And I think BS on that. It cannot be actively clustering in the sense that it's rerouting two axons so that they can be closer together. It's having to work with the random population of spines that and axons that it has to do the LTP.
Yeah, so a few things. I agree that even if they didn't see any like clustering, it was just the plasticity results from earlier, that would still be interesting and like a demonstration of the cool plasticity between the two. But I did wonder, and perhaps this is naive of me, whether you know, it is useful to know that you do see these synaptic clusters, in that like maybe that does have some implications for things you could find in tracing studies to try and read out memories from them. I guess a point against that they even mention in the paper is that if you look at the kinds of spines that they saw, they didn't see any difference in obvious morphology between the different spines and the different clusters. So they said the distribution of like thin versus stubby versus mushroom spines all look pretty similar between the different animals, the different types. But in terms of like active rerouting of spines, or sorry, of axons, I mean, it's true. I guess like I don't think the axons are necessarily going to be like, you know, rerouting themselves into clusters, but you will see presumably some degree of like actual spine growth and spine survival and increased densities in a way that's going to be notably structurally different in a way that like demonstrates that.
But those spines can only have an effect where they can reach over to the axon. So it's really just where axons and dendrites happen to be close already. That's the only place where you can even modify anything really that makes sense, that makes any difference.
Yeah. No, I mean, that's true.
The other thing, I know we're over time.
That's right.
The other thing I wanted to quickly say is that from a motor engram decoding point of view, the birdsong system is definitely on our radar as a memory decoding thing. And the main reason for that, at least I believe, is that there is this output to RA, I think it's called RA, I could be wrong, where there is an understanding that there's motor groups. And so there's some understanding of what the output would be. And I don't know if there's anything like that in dorsal striatum. If there was parts of dorsal striatum, like in the Tony Zadar paper, where you could say this is a lick left and a lick right, then that would be something to hang on to. Anything to hang on to from the input side or the output side seems to be almost a requirement for a memory decoding problem.
Yeah. Yeah, that's a fair point. When I was trying to imagine ways you could use this to help with memory decoding, I was struggling to think of something that didn't count essentially as cheating, insofar as like you'd have to have already known like this population of engram neurons in the M1 was involved in this task and this other population was involved in a different task, which sort of then defeats the point of then looking at how they're connected into the DLS in different patterns.
I think the amount of cheating, quote unquote, allowed is still open to, because I mean, if you have to label some stuff, but that labeling is getting to the ground truth, and then you show that you can decode other things based on that, and then even use that as a check. That seems to me like not a cheat, but actually a feature.
Yeah. I mean, I definitely agree with using it as a check. I just more meant like if you did something dumb where like you used the trap procedure, but with like 5 different colors, and you like train the mice on one task, used yellow, train the mice on another task, use green, change the mice on another task, use red, and then ex vivo, you were like, oh, look, it's the green ones that correspond to this particular task. That would be cheating, presumably.
Yeah.
That's more what I meant. Cool. Well, that's all I have from this paper. Does anyone else have anything? Anyone who hasn't spoken, maybe? Otherwise, thanks for coming. And we have another one let's look at my list. On
the 20th.
Yeah, on the 20th. That Randall are you still presenting that one, Randall?
Yeah, that's the plan.
All right, great.
Unless the authors themselves decide to. Yeah.
Well, that would also be fun. Otherwise, we will see everyone back in 2 weeks from now.
Thank you very much. This was awesome.
All right.
Yeah.
Thanks for coming, everyone. Speak to you soon. Bye.